My name is Howard Chan. I'm with the Biocom Institute and excited to be hosting this event today. Before we get started, I wanted to go over some logistics for this webinar. First, we want to let you know we are going to be recording this session and making it available on our Facebook community group after this event. Secondly, we will have a Q&A opportunity at the end of the keynote. So please feel free to post your questions ahead of time in our Q&A. And Marissa from our team will be monitoring it. You are also welcome to use our chat function as well. Lastly, we want to invite you to join our K-12 Life Science Biotech Educator Facebook group. This K-12 Life Science Biotech Educator group is a community space for educators to share resources, events, and connect with others who are passionate about life science and biotech education. We encourage you to share links, post questions, and comment in this group to help support the community. Marissa will be posting this link on the chat box here, and I hope to see some of you there shortly. And now I am honored to introduce our keynote speaker for today, Dr. Tomi Pastinen. Dr. Pastinen has been the director of the Genomic Medicine Center at Children's Mercy, Kansas City since 2017. Dr. Pastinen joined Children's Mercy from McGill University, where he co-led the Genome Center and supervised a multidisciplinary human genomics team from 2006 through 2017 as a professor of human genetics. He held a Canada Research Chair from 2007 to 2017 and has authored over 160 peer-reviewed publications, including papers in top journals, Cell and Nature Genetics. Prior to joining Children's Mercy, he led the Epigenome Mapping Center at McGill as part of the International Human Epigenome Consortium, which produced maps of human genome function to the global research community. Dr. Pastinen earned MD PhD from University of Helsinki, Finland, followed by postdoctoral training at McGill University, and was mentored by Drs. Leena Peltonen and Thomas Hudson, respectively. Currently, he is leading Genomic Answers for Kids, a foundational initiative shaping genomic medicine at Children's Mercy that aims to learn from 30,000 patients and their family members how to solve unsolved rare disease in children. Please welcome Dr. Tomi Pastinen.
Thank you, Howard. I hope everyone hears me well. I have to join through my phone, which may produce some technical problems, but I hope we get through this. So I'm really happy to be here and be able to share our work on finding answers for rare disease through genomic methods. And if we go to the first slide, I will first describe you a little bit of background on genomic risk in general for disease, and then I will delve into the rare disease problem, then describe current clinical sequencing approaches and gene discovery efforts, and finally discuss of the program that Howard just mentioned, Genomic Answers for Kids, which is a Children's Mercy health system-wide program to find answers for unsolved disease. Next slide, please. So in cases of disease, we have different levels of gene involvement and environmental influences. And what we are interested at Children's Mercy is particularly the diseases in the bottom part of this graph, where genes play a deterministic role and environment actually only tweaks the risk of disease. So if you have gene mutation in these type of diseases and two siblings, for example, share the same mutation, they have very similar disease course in general, and the environment really plays a small role in modulating the disease, and it's not usually involved in triggering the disease. If we go later in life, the influence of environment increases over time, and for adult onset, very common diseases, environment plays a predominant role. But the bottom part, if we go to the next slide, is really the rare disease. What we are trying to solve using genome sequencing And for rare disease, genome sequencing is the first line test, whereas for other types of diseases, genome sequencing can be used in research context, but it's not usually used clinically. So rare disease definitions vary across the globe, but a rule of thumb would be that rare disease is a disease that occurs in less than 1 in 1,000 individuals in population. So individually, each of these diseases is rare, but collectively they are relatively common. So 1 in 10 Americans or 30 million Americans is estimated to suffer from a rare disease. When we look at our population and children in general, 1 in 30 child is born with rare disease, and it usually takes up to 6 years to diagnose such a disease. And part of the problem is really the multitude of these diseases and the complexity of defining the disease. And I'll show you some additional problems. So many of the kids actually never get the answer, and families remain in limbo in terms of understanding why their child is ill. Rare disease is also characterized by several misdiagnoses across their diagnostic odyssey. And what we do know from rare disease is that despite their 1 in 30 incidence in newborns, they cause 1 in 6 hospital admissions in a tertiary care center like ours here in Kansas City. Most of the cases of rare disease that we are sequencing in our patient population are actually even super rare or ultra rare. They occur in less than 1 in 50,000 individuals. And most of these diseases have a name, but they don't have understanding of mechanism of the disease, and very few of them have any working therapies. But I would stress that regardless of the lack of therapies, finding the reason for the child's illness is always impactful for the family. They need to know why their child is ill, what is expected in the future, and how can they link to other families through peer support mechanism and social media to understand better their child's illness. So despite the lack of treatments, families are seeking for these answers to prevent further futile diagnostic tests and unimpactful treatments. So next slide, please. So how do we find these diseases? Traditionally, we needed to guess very carefully where in the genome we were looking for, and we used traditional genome sequencing as shown on the top of this slide, where we needed to decide from the 20,000 genes in the human genome, which gene would we sequence, and even more specifically, we needed to often decide which part of that gene we would sequence. So this is called traditional or Sanger sequencing. It won the Nobel Prize several decades ago, but it's completely inefficient in large-scale sequencing of unknown parts of the genome, because it would take millions and millions of dollars and years to sequence a single genome using these traditional technologies. So approximately 15 years ago, next generation sequencing that is shown on the bottom part of this slide was introduced, and this generates small pieces of DNA in a massively parallel manner throughout the genome from the patient DNA. And by comparing, if you click further, by comparing these short reads of the genome to the reference genome in black on the top, you can see differences to the reference genome, and these are mutations in the patient genome. And if they occur in the right type of a gene, they could be cause for the disease. So currently we can do exome sequencing, which means sequencing the parts that code for proteins, or whole genome sequencing, WGS, that sequences every part of the genome with some caveats, as I will show you later. So next slide, please. So clinical sequencing is carried out at our genome center here at Children's Mercy. Here are some practical implications. We do need to have a payer for the sequencing. So one part of lack of diagnosis is that some families don't have access through their insurance carrier to genetic testing. One big problem globally and in the US is that we don't have enough genetic counselors, trained personnel that can tell the family about the risks and expectations related to genetic testing. What we do hope when we do genetic testing is that we not only sequence the child, but we also sequence both parents. As you will see why that is very important. What we report out from these sequenced genomes is then pathogenic or disease-causing mutations and likely pathogenic or likely disease-causing mutations, as well as variants of unknown significance, which are very common in the genome because all of us carry many mutations in our genome, and sometimes we don't understand their significance. Overall, the diagnostic rate is around 25% in our diverse pediatric setting. The low percentage partly reflects the liberal testing. We are in a fortunate position at Children's Mercy that we can test a lot of our patients. But if we go to the next slide, we see that the diagnostic rate globally is only up to 40%. So in the best case scenarios, when the patients are selected very carefully to be enriched for genetic disease, particularly through family history, for example, even then the diagnostic rate after sequencing the genome is around 40%. So why is this? In some cases, the disease may not be genetic, even though we suspected it. Based on several studies, we anticipate that rare disease in general is about 80% genetic. So this still doesn't explain the lack of diagnosis in over half of patients. In some cases, we think that the genetic change is undetectable to the current clinical sequencing technology, and I'll show you examples of that. But in most cases, the gene is not yet discovered and we can't make statements. So these are remaining as variants of unknown significance or genes of unknown significance. So next slide, please. So in our hospital, the prediction of successful clinical next generation sequencing is really based on if we see a child with a very early onset, usually at birth, disease with severe global impairment, including things such as hypotonia or muscular weakness, or then later in development, we observe that the baby or infant is missing milestones such as sitting or crawling, as well as, of course, specific congenital anomalies in organ systems, in the skin, in the limbs, that may point to specific genetic diagnosis. And also, if we do have ability to sequence the whole family, both parents, that improves our chances of detecting rare disease mutations in the genome. But in the next slide, we go to the real challenge that we're still facing, is that despite the tremendous sequencing efforts throughout the community and every day in our hospital too, we know that many of the rare diseases still remain undiscovered and the genes underlying them are unknown. So we estimate we have about 4,000 genes that are known to cause human disease, and they cause about 6,000 different diseases. So the same gene can cause more than one type of disease. And the current rate of gene discoveries is 150 to 200 genes a year, and 100 new disease associations are discovered. So we could face many, many more years to discover the remaining rare disease genes in our genomes. And this means that many families will remain without answers for their child illness. So in the next slide, we introduced, due to these challenges, health system-wide program in July of 2019. So we are a little less than 2 years into the program, where we want to provide greater access to genomic testing and also provide new technologies from the research space introduced into genomic testing that will allow us to carry out higher diagnostic success through the years to come. In the next slide, we show what we are aiming to do. So in the top part, the situation today is shown where a small proportion of kids and families with rare disease are getting comprehensive genetic testing, whereas in genomic cancer for kids, we want to provide, regardless of insurance coverage, for example, genomic testing to all kids where their provider considers that there's a chance that this is a rare disease that is genetically caused. And over time, we want to improve the access, obviously, as well as improve the rate of diagnosis using the integrative genomic assessment. We are early on in our path in this program. We aim to collect 100,000 genomes from 30,000 families, and we've done a little less than 3,000 families to date in this program. In the next slide, I'm showing you a picture of one of our first patients that we saw. This is 4-year-old June. And in the next slide, we have a short medical history of June. So June had a difficult birth. She had significant respiratory distress, and she was admitted to neonatal intensive care unit about 5 years ago, 4.5 years ago. And at the time, at birth, she was noted to have several congenital anomalies, but there was no family history of disease. So the parents were apparently healthy. At the time, June was fortunate in the sense that she did undergo whole genome sequencing by the tools that were available and the knowledge that was available in 2015. But at that time, the mutation was not found. So she was one of the first ones that were enrolled in July of 2019 into our new program. Her clinical presentation had evolved somewhat over time, which is always important to remember that kids that didn't get, for example, diagnosis at birth may get it later on in life because there are some features in their disease that come apparent to the physicians and then help to interpret the genome sequence. So we go to the next slide. In June's case, we found the mutation that actually fit the evolved clinical picture in a gene called MYRF. And these colorful pictures in the bottom with the red box highlighting a little tick mark in those rectangular bars, which are sequencing reads. The little tick mark is the mutation that June has in one of her chromosomes, and the two other samples in the rectangles below the tick mark are parents' genomes that don't have that mutation. So this is a mutation that arose in June and is not seen in parents, and therefore is highly likely to be causal for June's disease, specifically as other families have been described with the same type of presentation with mutations in this gene. So we were able to end June's diagnostic odyssey of 4 years in only 2 months with the new tools we had for the program. So in the next slide, I'm showing you another example of a slightly different example. June only found the answer for the disease, but this disease that June has is very rare, and there's no potential therapies. Another very rare disease was found by our program in Caden and Ty that had also been suffering from abnormal fat accumulation in their upper body as well as their liver for years, and it had been genetically dissected before clinically, but it had not been deciphered what the brothers had, and we introduced them into our program and found a very, very rare disease. But we were fortunate that the features of this disease were investigated by a physician in Michigan that could instruct our clinical team on potential symptom alleviating interventions, such as surgical interventions to remove some of the excess subcutaneous fat, as well as then follow up these patients as a specialist in this very, very rare disease. So in this case, the rare disease finding actually led to actionable therapeutic choices for the kids. So in the next slide, I'm summarizing what we are trying to do globally for rare diseases. We scientifically want to develop through collection of this large amount of data across 100,000 individuals, a new recipe for unsolved disease. And it starts from the top part of that arrow, where clinical genome analysis has been negative. And what we want to do on top of clinical genome analysis, shown in different parts of the arrow below, showing different technologies. All are based on human genome sequencing, but sequencing the function of the genome or sequencing different parts of the genome that are not accessible by clinical sequencing today. So an example of that sort of change is shown in the or a case showing a new sequencing variants is shown in the next slide. So this is our first challenge in genome sequencing is some DNA sequences are not observable by the current short-read sequencing technology that I showed you in one of the early slides. So we had this large family. We can actually jump to the next slide. This is the family tree showing the kids in the bottom row. The boys are the squares and the girls are circles. And the dark coloring means that the kid is affected with the neurological disease that is characterized by several different features: dystonia, seizures, and ataxia. And this family had been looking for answer up to 7 years without any genetic test finding the answer. And clearly it's genetic, since there's multiple family members, and even the lighter colored parents that are siblings have some neurological dysfunction. So if you go to the next slide, in this case, what we applied, and if you click a little further, we applied third generation sequencing, which allows us to, instead of reading 200 base pair or pieces of human genome, we were reading 100 times larger piece of the human genome, which allowed us to see changes in the genome that are invisible to this short read sequencing that is today used in clinical sequencing. And this research technology led us to this gene called STAR-D7 that had just been described on the same month that we actually used this new technology to sequence these patients' genome. We go to the next slide. We found in all the family members, so 7 in total, that had neurological symptoms to carry an expansion of a repeat sequence in the intron 1 of the STAR-D7 gene that was not seen in any of the normal controls and was described in that article that I had in the earlier slide to be potentially causing this new neurological disease. And that's what we think this family has, is this new neurological disease that was invisible to earlier sequencing technologies. So if we go to the next slide, the second challenge we have is that in addition to missing some parts of the genome, we are not interpreting the whole genome. So clinical sequencing only looks at coding parts of the genome, which constitute about 1.5% of the genome. In contrary, we know that nearly 8% of the genome is actually evolutionary constrained. So it probably has a function, we just don't understand it too well. So we don't interpret that even if we sequence it. And the gray parts in the top arrow there are the parts of the genome that we don't assess, which include gene regulation, as well as RNA expression and protein expression. And what we can do now with different sequencing technologies, not looking at only the sequence, but the function of the genome, we can actually access these gray parts of the genome and start to look at the dark parts of unanalyzed genome that is not currently interpreted in clinical sequencing. And an example is in the next slide, where we used a technology called single cell RNA sequencing. We're using the same technologies to sequence, but now we're looking at RNA species from individual cells. On the right side graph, we have a normal single cell RNA profile from peripheral blood of a patient. And on the left side, we have a very rare disease called Roidman Syndrome that is caused by a mutation that is invisible to most clinical sequencing approaches today, but it causes a very distinct single cell pattern, which is indicated by these greenish dots in the upper left corner and with shown by the arrow. So this is an unusual single cell RNA signature that is caused by this rare disease. So instead of looking at the sequence, we're looking at function of the genome and finding these rare patterns that will allow us to interpret some of the unsolved diseases. So in the second last slide, I'm showing you the key part of what needs to happen next, which is data sharing. So we are working hard with many other rare disease groups to try to crowdsource genome sequencing data, both the DNA sequence as well as the functional data, together with the patient features to be able to do matchmaking across multiple different pediatric hospitals to actually be able to characterize new diseases, because a single patient with the single mutation is not sufficient. We need to find additional cases across the globe to make the definitive diagnosis that this is a disease gene. So this crowdsourcing is a really key area of development, and families want it, and it's required for new disease gene discovery. So to conclude, in the next slide, we are experiencing the revolution of genetic diagnosis through the next generation sequencing technologies in clinical use, but we still have a couple of hurdles to pass, both on technology side, but importantly in data sharing and understanding the parts of the genome that haven't been investigated in the past decades, which include the non-coding parts of the human genome. And there are some hurdles to this, but if the global community comes together with this in rare disease, as they did for human genome sequencing in general, I think we can pass those hurdles, and 5 years from now, the diagnostic rate will be above 50% and closing the gap of discovering all genetic diseases, which sets these families to a new path of potential for developing therapies, as well as understanding their kids' conditions better. So I'll end there with the acknowledgement slides of my team on the left in the Genomic Medicine Center, as well as my clinical partners in the hospital and international collaborators. And last but not the least, we have generous funding from the philanthropic community in Kansas City and beyond, as well as from funding agencies. Thank you, and happy to answer any questions.
Awesome. We have a couple questions here. So the first question that came up was, what makes certain mutations invisible to either Sanger or next-gen sequencing?
So most of the human genome is called single or unique DNA, single copy or unique DNA, but parts of the human genome is repetitive and not unique, but it still has function. There's still duplicated regions, for example, in the genome that have expressed genes, and these regions are very difficult for short reads because they can't orientate themselves well enough in the genome to be able to call genetic variation at single base resolution. So it's this, the repetitive nature of parts of our genome makes some mutations invisible. Some mutations then other mutations have actually a mechanism which is invisible, which is expansion of a region of the genome. Again, this is the expansion of the region of the genome happens through expansion of repeated elements in the genome that are invisible. So these are repeat expansion diseases that are increasingly missed and need to be specifically queried, not by next generation sequencing, but by other technologies. And if there are new ones, you won't find them. The STAR-D7 was one of the newest repeat expansion diseases that was described just a year and a half ago.
Wonderful, thank you. Another question that we have is, do you use a phenomizer?
Yes, well, we use multiple similar tools. We don't use phenomizer. We have newer algorithms, machine learning algorithms that we use jointly to understand. So phenomizer links patient clinical features to a specific gene that has been described in the literature. We use later versions of phenomizer, but the principle is the same. Extracting relevant information from patients' medical record and linking that with literature knowledge of gene mutations to create candidates. So it is that approach is very relevant to our genome interpretation.
Great, thank you. And are most of the mutations you've identified in regulatory or structural genes?
So nearly all mutations that are currently clinically reported are in structural genes, protein coding genes. They change the structure of protein, but that's not because that doesn't exclude the potential for regulatory mutations. It's just that our framework of clinical interpretation of the genome does not allow claiming pathogenicity or disease-causing features in the non-coding DNA. So that's why we have a long road ahead of us to discover what proportion in real life of the functional non-coding DNA is actually potentially disease-causing. But clearly, given that we're missing so many diagnoses, there is a good potential that there's a substantial fraction that is in the non-coding DNA.
Thank you. Next question is, what are some of the main obstacles in data sharing among rare diseases?
I think people often cite genomic privacy and patient autonomy and things related to ethical, legal, and social implications of genomics. But when you talk to rare disease families that are in quest for looking reasons for their child's illness, they are generally, and we embed actually patient families and patient advocates in our research program. They are overwhelmingly supporting data sharing in cases where it can advance understanding of their child's illness. It's a different thing of putting your genome online if you don't have anything wrong with your genome, let's say. But these families actually stand to gain from it, so they are for it. So regulatorily, there's no clear impediments for that. It's a cultural change that needs to happen through our scientific community, where we actually admit to us that we shouldn't hold on to the data that we generate in these large programs. The right and ethical thing to do is actually share across all the research programs, not put it all open online, but really with legitimate scientists working on rare disease, we should share the data openly. I don't think there's any real hurdles. Families want it. There are mechanisms to do it. Regulations and protections are in place that we can share it with scientific community.
Great. And another question is, the image of the hospital lit up at night is amazing. Could you share a little bit about the design, inspiration, and links to genomics? The stairs look like a DNA helix.
Yeah, so stairs look like a DNA helix, but the side panels are actually, that you see lit up, a reflection of the way we used to do Sanger sequencing. The time when I was introduced into a genetic laboratory, this is how sequencing results look like on a polyacrylamide gel, and these different rows of windows that are sporadically lit with these blue lights are actually representing different base pairs. So if you go from there's the 4 different bases shown at different floors, and then these tick marks describe which base pair is at which position. So you can read this from left to right or from right to left, and try to find the human genome sequence that is actually engraved. So there's a couple of patient sequences in this facet of the nighty facet of the building that I just sit in, and I'm waiting that the lights turn on.
Amazing. All right. Another question. What are your thoughts on CRISPR to edit these rare disease causing in human embryos? Is that the end goal of your efforts to determine the causative gene?
Well, I think any sort of therapeutic manipulation of the genome in embryos is a very controversial, and I definitely don't think that's the goal. I think we can look for ways to correct genetic changes in newborn children and children in general, using targeted CRISPR-based technologies in the future to reverse some of the effects of these disease mutations. But I think it's a slippery slope to start to edit genomes of embryos.
Thank you. We'll do another call for questions if there's any more. I think no one has typed anything in, but if you have any more questions, go ahead and do so now. All right, well, it looks like we are all done with our questions. I'll hand it back over to Howard then. Thank you so much.
Thank you.
Thank you, Dr. Pastinen, for sharing your time and expertise with us today. As a reminder, we will be posting this video recording on our Facebook group. And now I'm excited to introduce the CSR program manager from Illumina, Vanessa Light. Vanessa is focused on supporting STEM education and will be sharing about all the amazing Illumina resources focused on genomic education. Please welcome Vanessa Light.
Thank you, Howard. Let me just take a moment to share my screen.
And you're muted, Vanessa.
Now can you hear me?
Yes, we can. Thank you.
Okay. Sorry, I was trying to find the unmute button and I couldn't find it. So thank you, Howard and Dr. Pastinen. It's always incredible to hear the stories about how our technology at Illumina is brought to patients. And so it's really wonderful to hear, the work that he's doing and just the impact that he's making. And as Sharon asked about the building, it really is reflective around the work that they're doing, from the buildings to the doctors to the patients. So it's really incredible. So as Howard said, my name is Vanessa Light, and I manage our STEM education programs at Illumina, which is a life science company based in San Diego. And at Illumina, we believe that genomics will change everything. You just heard Dr. Pastinen talk about how it impacts these families' lives and these individuals' lives. And really from how we diagnose and treat cancer, rare diseases, to feeding the world's population. And that really requires a very diverse pipeline of future innovators. And one of Illumina's philanthropic focus areas is STEM education. And so by partnering with teachers, educators like you, we can work to achieve our goal of inspiring the next generation of scientists and engineers, innovators, to pursue careers in life science and to support teachers in feeling able and ready to be able to bring genomics into their classroom. And those careers can be science-based or non-science-based. So this brings me to the how. We talked about the why, now the how. How can we help you all bring genomics into your classrooms? DNA Day is coming up April 25th and is recognized by celebrating the completion of the Human Genome Project and the discovery of the double helix. And so to connect with learners around the world, we're celebrating throughout the month of April. And we have set a goal at Illumina to connect with over 10,000 learners through our genomic literacy programs. So we know, you all know firsthand, the pandemic has taken a toll on students around the world. And the Future is Bright campaign really aims to create quality hands-on experiences for students and our employees as well. So one of the really wonderful things about Illumina as a company is that our employees love to give back into the communities that they live and they work. And through that, we really do that through STEM education and through working with educators. So we hope that providing free resources for educators and learners, and that we can create a more equitable access to STEM education. So some of the things that we're going to be doing in April, this is our inaugural year of the Future is Bright campaign, and that is, again, all throughout the month of April, is really to connect with community partners. And when I say community partner, I mean a nonprofit museum, I mean a school, a student group, a Girl Scout troop, a Boy Scout troop. And so how can we be able to connect with those learners to be able to bring our technology, the excitement around science, the excitement around DNA and learning to their groups? And so we know that each group is different depending on the age, the classroom mediums, whether they're virtual or in person, and the comfortability with genomics. And so to cater towards each one of those classroom styles, we have a handful of opportunities, ways that educators can opt in to engage with our campaign. So there are a couple that are going to be just in April, and then there are some that are year-round. So I'll go through each of those to be able to share them. So the first one is the San Diego Festival of Science and Engineering. And while I know that seems like it's San Diego centric, this year, because of being in a virtual setting, this festival is available to anybody. And so we really wanted to be able to showcase San Diego and the companies and organizations in San Diego to teachers around the country. And so these, throughout the course of the week, April 24th through May 2nd, we will have a handful of activities. And as part of those activities, we are partnering with Skype a Scientist, which is a nonprofit organization that connects classrooms and professionals to be able to learn about careers. And so we are doing some specific ones around genomics. So our first one is actually coming up this weekend, and it is, excuse me, it is focused towards teachers specifically. So we want to be able to showcase all of our programs and be able to equip those educators to really bring this into their classroom. So that is this Saturday, and Marissa will be able to put in the chat the links to these sessions if you'd like to learn more and sign up. The second one is later in April, and that is the Life Science Career Panel. So we will be hosting a career panel talking about all the different types of careers that you can have at a life science company like Illumina, not necessarily only Illumina, but other organizations and companies that are in San Diego and around the world. And then that is really geared towards the high school age. And then the one on April 28th is one called Curious Genes. And that is going to be more geared towards elementary and middle school and talking about all the fun, exciting, different things around DNA. So as I mentioned, we are working with a handful of, when I say handful, 500 handfuls of schools around the country who have registered to participate in our The Future is Bright campaign to either do a strawberry DNA extraction or a career panel specifically for their classroom. And we worked through several of our nonprofit partners to offer those opportunities, and we heard an incredible response. We were overwhelmed with positivity and opportunity. And so we have, while those spots have been filled, there is still our strawberry DNA extraction step-by-step tutorial that you could bring into your classroom. And so that is on our STEM site, which I've put on the slide here. And again, Marissa can put that in the chat if it's easier. But it takes household products. I'm sure lots of you are familiar with it, but it's really talking about what is the application of DNA and genomics, and how can they see the science that goes into this, and how could they apply that to whether that's moving forward in more complex science. So the next one I'll talk about is called DNA Decoded. And DNA Decoded is a set of curriculum that was created by our partners at Discovery Education. And all of these, we have 12 different pieces of content that are next generation science standard aligned, and they talk about precision medicine. So, when you're thinking about Dr. Pastinen and ways that you could apply this. So if you were to take the precision medicine content and then take the learnings that you learned from him today, and to be able to bridge those two and bring those into your classrooms, that's a really unique opportunity for you as an educator. And you don't have to create the curriculum, which is such a burden on time. So I will share the different opportunities through DNA Decoded as well. The last one that I wanted to touch on is our Lumina career videos, which also is a really nice supplementary material for those DNA Decoded curriculums, and also to make these career conversations, really relatable. And so this is a set of career videos that are talking to our employees and really getting to know them. Who are they inside and outside of the office? What makes them tick? What makes them continue to be learners, lifelong learners? And so those are all on our STEM page as well. And they come with a set of career cards that ask the questions around, what kind of classes should I take? What are some of the hard things about my job? What are some of the wonderful things about my job? So, so that is the kind of our different resources that can be made available. So on the next slide, is talking about our social media toolkit. So I'm going to go back for 1 second. But when you look at those April activities, if you are wanting to do those and share those through the festival or Skype a Scientist or the DNA extraction, we encourage you to be able to share those on your social media channels. It makes it fun, engaging, it makes it fun for your students. So many of the teachers now are on Instagram and Facebook and ways to share the different content and ideas that they have. And so, so we have this social media toolkit that has cards that you can post to social media and frames, so you can see the Genomics for All frame, and that is the hashtag that we're using for this campaign. This Future is Bright campaign is hashtag Genomics for All. So if you post anything, tag Illumina on the different channels that you see here, and then use Genomics for All. And so with that, I'm going to pause before I talk about one other opportunity. But I'd love to see, just to get a sense from the teachers, if you can answer this question for me. And one of the things that we hear a lot from teachers that we work with is, I'm familiar, but I just don't, it's really hard to be able to use the applications or understand the applications. And the technology moves so quickly. How do I keep up with the technology and keep up with my curriculum and things like that? And so that's really where that DNA decoded piece comes in. And so that you can be able to leverage those different applications that we use and not have to take the time or piece it together like so many teachers do. So, great. Well, looks like we have a little bit of a split. Definitely more intermediate, which is why you're all here. And then let's do so 6 of you are newbies, 22 are intermediate, I would love to know more, and 11 are advanced. And then the second question, now that I've covered DNA Day, is do you historically celebrate or recognize DNA Day on April 25th or within the month of April? And it looks like the majority, oh, a couple more coming in. So the majority of you said that you don't recognize this, which is really, I think, an opportunity for us to be able to connect with you all and be able to share ways that you can do this in an easy, approachable, kind of bite-sized way. So the last opportunity that I wanted to share with you all really comes back full circle to Children's Mercy Research Institute. And so they have a decoding challenge that we have partnered with them on called DNA Decoding Challenge. And up from now, it has launched. And from now until April 29th, so the month of April, they are inviting all high school students, student teams, classrooms to participate in a real world learning activity, to take sequences that are displayed on those outside windows that you saw. Those red tick marks represent 4 different patients. So those 4 different patients, there's a set of activities that goes with each of them that's in this activity book that's on this slide. And so it's really an open challenge to, and an opportunity for you as teachers to be able to share these real world connections. And again, it's a great way to, if you talk about precision medicine and then these real world applications. And then on April 29th, they will actually be doing a virtual town hall where you can speak with some of their doctors and their heads of the hospital. And so to better understand the work that they're doing. So it's a great opportunity for you to be able to connect with your students, give that opportunity to them. So I encourage you to, I will send the link out for that to everyone who has attended, and I encourage you to participate. So with that, I will go to Q&A.
All right, we have a few different questions here.
Yeah.
So I'll combine one of these. So I'll combine some of these, but would Illumina be able to provide NGS or multiplex sequencing for students working on capstone projects?
So we have the short answer is no, we wouldn't, but they're like from a sequencing perspective currently. We do have a program called Illumina Genomic Discoveries through Biocom Institute as our partner. And we train teachers to be able to bring this opportunity into their classroom. We've piloted it in San Diego and are hoping to expand that. But from the opportunity to connect with some of our employees, possibly from a professional lens, to be able to get feedback on their projects, I think that would be, if that's of interest, that would absolutely be possible. But as it stands right now, we're not able to run those tests for capstones.
Great, awesome. Would Illumina be able to provide Sanger sequencing of DNA barcode amplicons for school projects?
So I think that goes into the same question from before.
Perfect.
There is more specific questions. I'm happy to, or brainstorming with the 2 individuals that asked those questions. I'm happy to brainstorm to see how we can support if it's not necessarily from a sequencing perspective. So know that conversation we can definitely have that conversation.
Awesome. Would Illumina consider donating any isothermal amplification platform equipment to a high school?
So it depends on what it is. I think we have a possibility of, it really depends on what it is, and if there's kind of surplus equipment that our labs are getting rid of. So if there's a match, again, I think that's something we can either go through Biocom Institute or, or we can connect. I feel like I keep saying no, but it's
there's more specific details necessary in order to facilitate that process. i.e., feel free to contact us directly. I think you have many of our email addresses, so that, and you will, if not after this, after we send out a recording this week, and we can see what we can arrange to provide your students with additional resources or equipment or whatever that might look like. Okay, so next question is, the hashtag Genomics for All really glosses over a lot of the ethical issues in modern genomics. Are some of these issues explored within the curriculum?
Yes, so we have a curriculum that's around race and genomics. specifically that, and when we were creating that with Discovery Education, we were a little bit, I as a just individual was a little bit hesitant to put that out there. And the reason why we did it was because we were getting that question specifically from teachers, that they wanted to be able to have this conversation with their students. And so with that ideation and that permission from the teachers of wanting to do that, we wanted to, again, support the topics that you all want to bring into your classroom. And so, so I think that's a really good opportunity, and it talks about, it talks about race and genomics and ethnicity. And so, and also guide some of those conversations. So that definitely is in that, within the DNA Decoded.
Awesome, which the link is in the chat box there. If you find it there, you can find it there. All right, next question is, can the decoding challenge be done on Chromebooks? Yes, it can. So that activity book, we can send that out. We'll send that out in the email with the survey, or not with the survey, I'm sorry, with the recording. But it's a PDF, and then it links you to a Google Doc where you can submit all your answers. So everything should be able to be done on a Chromebook. Awesome, thank you. Next question is there an anticipated time required for the decoding challenge? Packed curriculum makes it difficult to add in more.
So I think the nice thing about the decoding challenge is, is that it can be done a bit of piecemeal. I think I would have to check with the Children's Mercy team on what they anticipate that time commitment being, and can let you know. But I would say it's, again, you have from now until the end of April. So if you wanted to parse through that, and but I don't anticipate it being a very long challenge. But there's more information in the PDF.
Awesome. And I know Sharon just popped in here to say that if anyone is specific, has any specific donation requests, I'm going to pop this into the chat box to please contact csr@illumina.com. And I believe that is all of our questions. And I know that we're here on the hour. We would absolutely love your feedback on today's session. I'm going to pop a Survey Monkey link into the chat box. If you don't mind providing us with that feedback, that would be fantastic. And otherwise, we will send it out by email with the recording in the next couple of days. But thank you for joining us. Thank you, Vanessa. Thank you, Howard, and everyone who attended today. We hope that you learned something and enjoyed it and have more resources to take back into your classrooms. And we are here for you guys as well. And Howard just popped also the Facebook group again into the chat box as well. So definitely another way to connect with us.
And I'm happy to stay on for a couple more minutes if somebody has another question that is more specific to them.
Thank you, everyone.
Thank you, everyone. Have a good evening.
Thank you.